Welcome, everybody. We are going to go ahead and get started in our January 22nd webinar, Insights in Biology, with Dr. Kay Aull, our USABO alum from '03 and '04 and gold medalist. I'm Rebecca Carter. I'm the new program manager of the STEM Lyceums program, brought to you by the Center for Excellence in Education. I am joined here today with Dr. Shannon Tyler, the program manager of the Teacher Enrichment Program. So we're going to go ahead and dive into today's presentation. While listening, I encourage you to take notes. So some background about the Center for Excellence in Education. It was founded in 1983 by Joanne DeGennaro and the late Admiral H.G. Rickover, who was the father of the nuclear navy and civilian uses of nuclear power, with a mission to nurture high school and university scholars to careers of excellence and leadership in STEM. Our mission here at CEE is to nurture high school and university scholars to careers of excellence and leadership in science, technology, engineering, and mathematics, STEM, and to encourage international collaboration between and among leaders in our global community. We are fortunate to have quite a number of programs here at CEE, starting with the Research Science Institute, or RSI, which is a highly competitive 6-week summer and science engineering program open to rising high school seniors, and juniors are also eligible to apply. RSI is the first cost-free summer science and engineering program that combines on-campus coursework and scientific theory with off-campus work in science and technology research. We also will hear a little bit more about USABO, or USA Biolympiad, the premier biology competition for high school students in the United States. It provides the motivation, curricular resources, and skills training to take them beyond their classroom experience to the level of international competitiveness. Since CEE began administering the USABO in 2003, every member of the USA Biolympiad team has medaled at the IBO. Next, we have our Teacher Enrichment Program. TEP provides opportunities for middle and high school teachers to connect with leading experts from industry and academia, explore cutting-edge research and development, and make meaningful professional links with direct benefits for teachers and students. Finally, we are here currently in our STEM Lyceums program, which is our virtual club for high school students to meet once a month and learn about STEM career pathways from leading field experts. Industry professionals share cutting-edge research and offer unique learning experiences like scholarships and internship opportunities for high achievers. Please visit our website, www.cee.org, to learn more about our programs and opportunities. And I believe Dr. Tyler will have dropped in the chat our links to our web pages. Now I will begin to introduce our speakers. So Dr. Kay Aull, USABO '03 and '04 gold medalist, started with a childhood dream of building things of biological parts. She learned as much about biology as she could in high school, then went to MIT, becoming the first to receive an undergraduate degree in biological engineering, with some help from alphabetical order. While working in a variety of labs, she found herself consistently drawn to the fun part, finding patterns in the large and messy data sets that biology tends to produce. She followed that impulse, doing a Ph.D. in Bioinformatics at UCSF, and is now working in a special projects group at Thermo Fisher. Thank you to Dr. Aull for joining us today. I'm going to go ahead and let you start off your presentation, and afterwards we'll have our Q&A session.
Hello, my name is Kay, and 20 odd years ago, I was in high school and I was competing at the IBO. And now I'm a working scientist. I do bioinformatics and I like my job and I'm excited about it. And so I was invited here to discuss the sort of things that a bioinformatician actually does and what I did in order to get from the IBO to where I am now. All right, so this is a slide from the actual job talk that I gave to get my current job. And this is a schematic of how I understand my role and what bioinformatics is to me. And so my job is making things with biologists, and the biologists, they have a question, they have things they want to know, but we have outgrown the era where you're doing this blot by blot and base by base, hand making these individual experiments. We want to do experiments at scale, right? We want to look at all of the genomes, all of the transcription factors, all of the cell types. And that is going to take, well, it's going to take automation. We're going to need to use all of this technology that we have now. And this is going to generate large data sets. And this is not necessarily something that the biologists are trained to think about. Hope you can't hear my hairball meowing in protest. So you have these piles of data, and hey, you need somebody who is trained to help you deal with that, because a pile of data is not an answer. And so you need an analyst, someone like me, who does this for a living, in order to actually look through this hairball of information and say, okay, here's where the patterns are. Here is the information that you're looking for, to be able to structure it in a way that humans can understand and act on. So that's my job. That's where I come in on this kitty right here. And so, if more broadly or more formally, what bioinformaticians can do in the real world, they do a lot of data analysis. So any sort of large scale biological data sets, you got genomics, you got proteomics, transcriptomics, you can use statistical methods, you can use computational pipelines, all sorts of analysis software in order to identify these patterns and trends. Sometimes, if the algorithms don't already exist in the world, you're going to have to develop your own. What is the best way for me to process this data in order for me to go from the hairball to the answer? And so you're designing these methods, and you're going to be implementing them in order to solve this specific biological problem. The famous ones, you got sequence alignments, you got protein structure predictions, you got gene functional predictions. Does this mutation actually matter? That sort of thing. Or it can go down to really bespoke sort of custom analyses to address whatever you're interested in. And this can involve, there's maintenance work, providing that sort of infrastructure to be able to look up the answers to your questions. Somebody's got to create and maintain these biological databases. Like, okay, here's all of the genes, here's all of the gene ontologies, here's all of the protein structures. And doing all that annotation and curation of data, there are people who do that for a living. And it's an important part of just being able to store up all of the data that we've gathered and reuse it. And also software development. Somebody's got to make all of this stuff usable. You write anywhere from scripts to like actual GUI packages in order for people to come in and analyze their data, even if like maybe they're biologists and they want to analyze their data without having to go knock on the door of the bioinformatics course. Like, I just want to know what cycle my qPCR stopped on. Can you do that for me? Somebody does that too, and that somebody's a bioinformatician. But a lot of it is collaborating with researchers and making things for biologists as well as with them, helping them do their job and helping them get to those answers. So I was asked to address, how did I get here? I was a bit of a weirdo. I picked my career at like age 7. And so how I got here, I was reading one of those little Discover Pop Science magazines, and it was talking about how in the distant science fiction year 2000, this was in the very early days of genetic engineering, when we rode brontosauruses to school, but we're going to do all of these things with genetic engineering. And they had this project where we're going to make edible vaccines, and we're going to make these suitable for countries that don't have the health infrastructure to keep their vaccines chilled. And so we're going to have these bananas, which grow well in the tropics, and we're going to genetically modify them so that they express polio antigens. Grow these bananas, and you take them to market, and you eat them, and there you go. And by the year 2000, like, okay, all of the biology Olympiad kids, it's going to be a short answer question. List me half a dozen reasons biologically why that's a bad idea. But it was the 90s and I was 7. And I thought that was the coolest thing I'd ever heard. So I'm going to learn biology. And I went hard. And yeah, that ended up with 2 IBO medals. And I learned biology, but it wasn't good enough. I figured out, okay, it's 2003. I'm applying to colleges. I still don't have my polio curing bananas. Why not? So the answer was, we don't really know how to build things out of biology. It's not an engineering discipline. If I have my polio vaccine banana, it's not going to help me get a measles vaccine apple. I'd have to do 95% of that work from scratch. It's all done as this little hand-curated, bespoke art project. We are not taking in the lessons from engineering. We don't know how to do these, you want modularity, you want standardization, you want to be able to reuse your parts. You want biological Legos. And so that was what the discipline of synthetic biology was talking about. And in 2003, the place to go to study synthetic biology was MIT. So I went, and I was in the first class of biological engineering majors. Last name starts with A, so I did actually get the first degree, but I was in the first class. And yeah, I learned how to do that. But I was also class of 2008, and the economy was kind of on fire. So I did synthetic biology, but I did some other things while I was kind of wandering around in the wilderness, attempting to keep roofs over my head. I like sleeping indoors. So one of the things that I did, I got involved with this group called DIY Bio, which is kind of an amateur biology group. This is a photograph of my closet in Boston. I've got this little genetic engineering lab in there. I did some basic bacterial transformations and DNA tests. This photo was actually on the front page of the Wall Street Journal. So yeah, you can do a little genetic test in your own house. And I also, I found myself for paid employment, I ended up working in a cancer lab. I was doing, on paper, I was, doing scrubbing the incubators and stuff. I was also the department sysadmin because, yeah, but it did give me exposure to kind of another side of biology that I had not worked in before. It's this human's cancer and seeing how they do things over there and learning that side of the bargain. And I was still, thinking about, why are we still so bad at building things with biology? And so what I realized was that, okay, we have all of these working biological systems. I mean, trees, plants, they work. How are they doing what they're doing? We as humans don't know how to build things out of these little biological parts that have their own specific properties. They're not bricks, they're not microchips, but Mother Nature does. What are the patterns that occur in nature? So I went to grad school to do systems biology. And so I can show you a little bit of what that looked like. I knew I didn't want to be like the cancer biology professor that I was working for, who had, he had his favorite transcription factor and wanted to know everything about it. I wanted to know, what is this as a part? So I was looking at trying to address some pretty simple biological questions. Like how does gene regulation actually work at the mechanistic level? You've got your favorite transcription factor and it activates your favorite gene. Okay, so we understand what this means at the cartoon level, at the population level. What does this mean at the level of an individual promoter? So I'm not going to go full research talk, but I wanted to show you just enough to kind of see what can you do with this side of bioinformatics and so, all right, cartoon level, my favorite transcription factor, your favorite gene, you are trying to activate it. At the lower levels, you have close to a linear relationship with how much of this activator is present. So one copy of my favorite transcription factor, making your favorite gene, it's linear, but at some point you're going to get saturated and you'll get this sort of saturation curve where, just can't, there's no more production available no matter how much of this transcription factor you have. So you end up with one of these hyperbolic curves for concentration versus activation. And we know this approximation works really well for like prokaryote bacteria. Fine. You're working with high copy plasmids, especially there's hundreds of these promoters per cell. You don't need to know anything else. This is your curve. But in eukaryotes, genomic promoters are only present at like 1 or 2 copies per cell. So the output of each specific promoter isn't lost in the averages. Can you still get away with this approximation? And the answer is no, not really. So to address this question, I worked with HIV. It's this really simple virus. It has one promoter, one transcript, and that transcript encodes one activator, which comes back around and turns this thing on. We know, based on this is how it works in the real world, that HIV has this ability to form latent infections. It can go into the genome and it can hide there for decades even. And we have equivalents in the prokaryotic community. We have lambdaphage, which is also a latent virus. Lambdaphage is 5 times larger than HIV. It's a lot more complicated. There's half a dozen Nobel Prizes for figuring out exactly how lambda works. It is a lot more than one promoter, one activator. Why? HIV is too simple to work, but it does. The general assumption is that HIV is taking advantage of the greater complexity that exists in eukaryotic gene regulation. so it doesn't have to hard code the bistable switching in order to just, you know what, I'm going to stay off for a couple decades. That's fine. So eukaryotic transcription, what you need to know about it is it's bursty. It doesn't behave in this sort of hyperbolic manner. Transcription occurs in cycles, a burst of activity, and then it quiets down. And so each of these plots, not mine, these are single cell expression over 60 hours. These are like large multi-hour bursts going on. These cycles take a better part of a day. We don't know why this is here, but it is conserved yeast to humans. And we have what parameter is this affecting? We have different parameters for the bursts. Is it increasing the rate of turning this gene on? Is it increasing the rate of turning it off? Which would, are you increasing the burst size by turning it off less often when the activator is present? Are you just expressing more when your activator is present, so you're increasing the KM, the production rate? Or are you just making it more often, bursting more often? So in order to measure this, set up this simpler system where I can induce my TAT, that's my activator, and then I can look at the output because it's nice and red. And I can do some single cell analysis and looking at this over time, do a little reality of what bioinformatics is like. I built a nice little automated image analysis program that was supposed to segment out these cells. But in order to test it, I went in and I clicked on 10,000 cells, the center of 10,000 cells times 120 images. And then I sent my image analysis program after it. To my profound annoyance, I was better at it. So I had to go and do the other 5 experiments. Click, 10,000 cells by 120 time points. That's why P in Ph.D. stands for patience. It really is the patience having degree. You're going to need some of that sociopathic level of patience to get through it, but the results, you can see 2 representative traces, cells off, and then it's on. It's off, on, and then it turns off for a short time in number 32, and then it comes back on. And I built the whole inference program to figure out when the mRNA levels are increasing and when they're not. And the result is that specifically the only parameter that cares about the amount of the activator that's present is the duration of the on state. So as you go up the rainbow, you're going up in the TAT activator concentration, and the length of time that you go in the on state increases. So this is the number of cells that are still in the on state if you start them all at 0. And so this suggests a model where what the activator is actually doing is it's creating a kinetic trap for this live expressing LTR. So if the TAT activator is present, it gets stuck in this on state. It can't actually turn off from this on state. It's just going to stay here and keep expressing for a longer period. It's going to stay in that state only when the activator is present. That was my PhD. And that is one of the things that you can do. Complicated problem. You need to apply technology, you need to apply analysis, but really this is the only sort of way that you can address a question like that for looking at, in this case, dynamic behavior. All right, that was my PhD. But coming into the end of 2016, like, okay, who's going to pay me for this? And so that's why I got back here. I realized, okay, I've been chasing this question, this question of how you build stuff with biology for a long time, but really, when you put me in a lab, what I want to do is look at these complicated data sets and figure out how to make sense of them. And that's what I'm good at, and that's what I want to do. So that's what I should do. And so I went out and I got a job doing that. And I work for now I work for the Thermo Fisher microarray group. Put this here. Hi, Thermo Fisher lawyers. You approved this talk in 2019. I can prove it, but I can show you a little bit of what that looks like. I mean, people think about microarray data as kind of this. It's an older technology. We all know that. And so you've got your panel of SNPs, and you're looking for genome-wide associations. If you find a really good one, you get a Nature paper. And if you find the kind of mid one, then you don't. But this is the sort of thing that you see, or you'll see in some database somewhere. Okay, I'm interested in this specific mutation. What's the population frequency? in various populations. Some of this data is microarray, some of it's sequencing, but somebody has to do the analysis work. What this looks like in real life is something like this, and somebody that's my job has to go in and figure out, first doing all of the data correction to turn this into something useful. This picture is back from 2011. We are now 2 generations of miniaturization and assay speedups beyond this, so the data it's worse. And we're also trying to make it do more and more complicated things. Yeah, and that is my job. I get to figure out how to do all the data corrections to make it into something useful, which, you're taking these little 2x2 pixel spots and you're turning them into, signal that you can actually genotype. This is what the raw SNP data looks like once you've gotten it processed out. You have the y-axis is the total intensity of binding, and then the x-axis is the contrast between the 2 different channels. It'll bind in one channel if it's the A SNP, and it'll bind in the other channel if it's the B SNP. That took slightly longer than I thought, so I'm just going to power through this and get rest. But yeah, the idea is I make it do more and more complicated things, trying to figure out how to make SNP genotyping work if we're not just looking at simple GWAS SNPs, if we're looking in regions of genomic complexity, like you have a recurring deletion, or you have pseudogenes, or you have other things that unfortunately some SNPs of interest tend to be located in, and just trying to extend the life of this technology, which, again, it is older than I am, but there are advantages to having much of your answer cheaper and for less analytical cost. Instead of having to sequence these things from scratch, you're looking at your panel of 500,000 curated SNPs that you understand how to interpret. And much of my work is trying to figure out how to add reliable SNPs to that portfolio with some analytical tricks, like splitting out these clusters by what the underlying copy number state is. All right. And so the other thing that I was asked to talk about is sort of what is the practical, what are some practical things that you should know about bioinformatics? And so first one is, what are the applications that I'm excited about for now and for upcoming in the future? And so I think the first thing is just the amount of data, and that has been true for much of my career. It's like we can do stuff now that we just couldn't have addressed before, because we have the data to do it. And that snowball has only kept rolling down the hill. It's getting bigger and bigger. So specifically, some of the things that are coming online right now: deep sequencing. It has become cost effective to use that in clinical settings. And this is hit the market within the past 5 or so years. You'll have things like non-invasive prenatal testing, where they'll just take some blood from you, and some small percentage, if you're pregnant, is going to be from the baby, and you just sequence it, and you can use your computational methods to figure out, okay, which of these sequences are from the baby, and you can use that to detect specific mutations. You can use that to detect chromosomal aberrations, like this looks like Down syndrome, or this looks like trisomy X, something like that. And you can do that very early in the pregnancy, like well before something like amniocentesis, which involves needles where you don't want them. Right now they're selling, like, at 6 weeks pregnant. That's barely a missed pregnancy test, but you can get your little blood draw and you can tell what sex the baby is. That's bioinformatics. That's a genuinely hard problem, trying to figure out what of this small percentage of DNA actually should be assigned to the baby, and whether that's enough information to be able to tell what's going on with that baby. And even more challenging is they're also doing this for cancer. They're trying to do it as a screening method. Hey, you're healthy, but we are looking for this panel of oncogenes, and if we find them in you, you've probably got a problem. You can do it for cancer surveillance. It's like, hey, you've got a stage 1, stage 2 operable cancer. We're going to take it out and we're going to sequence it to figure out what mutations are driving it, which was a big thing back in, the back in my earlier career, like circa 2010. Targeted drugs, that in itself has been an enormous contribution to cancer care. But now they're saying, okay, we can not only sequence your tumor and figure out what the driver mutations are, we can keep sequencing your blood and we can track how much of that tumor burden is left. So we know if it goes to 0, you're probably okay. If it's still puttering along in there, that means you have a metastasis somewhere. Maybe we can't find it, but you need to be treated more aggressively because you do have it somewhere. And you can use that to kind of save people who are relatively low risk from aggressive chemo and give that to the people who actually need it for their care. And so that is definitely something I'm excited about. I'm excited about, like everyone, I'm excited about, what can AI do? I'm so much of what I do and what many bioinformaticians do is pattern matching. Can you have help? Can you get the computer to kind of recognize some of these patterns? And we do, like in my job, I sell things. Part of my job is to try to figure out what mistakes the customer made. Basically, they all have different phenotypes, and we are training an AI to look at their pictures of their results and say, I think you did this, which is currently done by skilled humans. And that would free us up to do, more interesting things. So those are the applications. And also, next talking point is ethics. And that is, that is something that comes up a fair amount because we are, at least in my case, I'm dealing with humans. These are people. This is their genetic data. This is their health information in many cases. And so that is to be taken seriously. And also there are rules. There are regulations that we have to follow. In many cases, if we are not dealing with US customers, that the genetic information of their citizens cannot come to our soil. It has to be processed with them. So I have to log into servers that are physically present within the country where that data was generated and do whatever checks we need to do. If we need to help them, we'll do it there. That's a common data protection issue. And we also have the occasional cultural sensitivity. So I work on doing, again, I work on human arrays. We're doing genetic screening for reproductive health for people who would be at risk of having genetic diseases in their babies. And we designed this product under the Western model where women receive most of the reproductive care. And so we hard-coded the assumption that most of our samples would be female and that we'd only test the males if the woman had the problem. We've had a request from some of our customers, we want to screen the men first because culturally it would be stigmatizing for the woman if she was at risk of having a diseased child, even if it's a rare disease and it's not a problem unless her partner has it. So we want to test the men first. They're like, okay, that makes perfect sense. And we also have a lot of rewriting to do. Oh dear. So it is very much something that you have to think about, that this product exists in the real world and it will have real world implications. And so career insights. What do you actually need to know in order to do bioinformatics for a living? No matter what side of the spectrum you're going to be on, whether you're a pure biologist or like a pure statistician, mathy person, you really should know how to code, at least a little bit. The most common languages, it's R and it's Python. Python is a great first language. It's a good general purpose language. It's never going to be the fastest, but it's at least at the level that most bioinformaticians who are doing this sort of scripty ad hoc analysis work on. What matters is but in seat time, developer time, and you can just throw more computational resources at it if you have to. What matters is doing the development, getting it right the first time, because you're not, burning through the script over and over again. You just need, you're writing it once. And just the ability to do some basic scripting on your own, even if you're a pure biologist, it's going to save you a lot of hassle. I remember I was actually quite sad back in the day when Excel lifted its size limit. It used to cap out at 64,000 rows. That was a safety feature because that kept the biologists from analyzing their data in Excel. When it got to the point where Excel crashed, they'd complain to me. They'd be like, yeah, let me help you. You should be talking to me. Yes, in one case, I got one of those complaints. Just trying to analyze her RNA-seq data in Excel, and at some point, it sorted the columns, and the columns had gone their separate ways. And she's really excited because half of these tumor samples had this giant deletion in chromosome 10. The deletion was in chromosome X. So yeah, half of the cancer patients were male, but you don't analyze your data in Excel. X is not the same thing as 10 Excel. And the other thing that you should need to know is that many bioinformatician jobs don't actually require you to be excellent at math. By MIT standards, I am not. I do not have the makings of a research mathematician. I am perfectly fine when applying other people's math, but I should not generate new math for a living. And there are people who do that. They are legitimately mathematicians, but they are developing new methods for bioinformatics, and they're doing this as a rigorous math problem. Mostly like the academic side bioinformatics researchers who are genuinely pushing the field forward in terms of mathematical methods. But for people like me who are using it as a tool, it's okay that we are script kiddies. We use the math that other people have set down for us. And what you really need to know is you need to know what tools are out there. You need to be able to match these problems with the solutions, and you need to be able to communicate them with people who are anywhere from pure biologists to pure mathematicians. That's what matters. And the next topic that I was asked for is, so how did Isabeau come into this? So in high school, I don't know if I appreciated quite how useful it would be. I was really excited just to have the recognition, the opportunity. I was jealous of my math and physics friends. Like, why don't I get an Olympiad? But 03 was the first year that the US participated in the Biolympiad. Like, I finally get one. I got to go to camp and, assorted camp shenanigans. That's me being Miss Biology 06. But really, it was more, I was happy for the recognition. And I remember things like back in 05 when I was a camp counselor, they told us the practical was written, and I found out 18 hours beforehand that was a lie. So yeah, there was scrambling. There was catching insects under the streetlights at like 3 a.m. Once I was finished with high school and I moved on to college, the next most useful thing that it did for me was credibility. I could I could put it on my resume and say, hey, I can tell you that I know biology, and you can believe me or not. Say, you know what, I was one of the top 4 biology students in the US, and I've got a silver and a gold medal, so you should hire me. Credibility is good. And people did. But where it really became useful to me was once I'd gotten out, like out in the real world. I realized that the breadth that I had was actually like a big feature, because when I was looking for a job in those lovely post-economic collapse years, I got a job in the cancer lab basically because of Isabeau. One thing that sometimes happens in interviews is that, and it happened there, is they'll hand you a paper. So the cancer lab handed me a paper all about their favorite transcription factor, and gave me 20 minutes to read it, and then I had to present on it. It doesn't happen every time, but every single time that somebody has done that to me, I've gotten a job offer, because I did not have any pre-existing interest in MITF, their favorite transcription factor. But you know what? I had a basic context for everything that was going on in that paper. And so within 20 minutes, I was able to understand what was going on and make a little diagram and talk about it fine. It's also, there was a dissection component. Part of my job was preparing skin samples for research. It was a dermatology lab. How do you feel about dissection? Like, yeah, I'm good at dissection. Big bag of dead animals day at Biology Olympiad. That's a thing I can do. And so simply having that experience is a real advantage. And another example is that in the early stages of COVID, my company was trying to make a array for genetic predisposition. This is back in April 2020. And literally half of the human genome had been tagged by somebody as a potential COVID risk factor. And we can't put the entire thing on. So my job was to look through the gene ontology tree and decide which of these things I actually believed, tag those for inclusion onto the array. And that was, again, all biology Olympiad background of, okay, what I know about the immune system, I know about these, these things that they're talking about here. And I was, the only one in the group who could realistically do it. So the sheer breadth that you get from biology Olympiad is a big deal. As a bioinformatician, you do have a tendency to wander. You'll never know what's going to be relevant. We do all sorts of things. We don't just do human at my company. It was like, oh, here's a pine tree array. Okay, I know about pine trees. So next, how did I get started in research? Early research experience. I did do some research in high school. I'm hesitant to recommend high school research because it is really heavily dependent on who you know. Like, it's hard to get people to take you seriously as a high school student. You're a minor, you're a liability, you're probably just going to blow stuff up. Yeah, it's really difficult. I was not able to find a research experience through, just applying for them. I spent the summer of 2003 doing SARS research, but that was because my dad's college roommate was a professor doing SARS research. If you got it, flaunt it, but honestly, it is difficult, and I understand that. College, on the other hand, yes, you should be doing all of the research. If that is an interest of yours, if that is somewhere that you want to go in life, you really want to be doing that. And you are not anymore just a little nuisance. This is like actually part of the professor's job. It's part of what they're paying for. You should be doing this, and it's work experience. It is never going to be easier for you to get that experience and build up your resume and say, you know what, this is something that I know how to do. You should hire me for it. And you should know for yourself whether research is something that you want to be doing and what kind of research that you want to be doing. And you won't know that until you've actually gotten in there and gotten your hands dirty and tried it just to figure out what it's like. And research, it is not for everybody. Like, it is really different from sitting there and reading Campbell, where all the details have been worked out and everything makes sense. Like, you're the one thumping your head against the wall, trying to figure out what's going on. And so there's kind of a stereotype for grad school applicants, like, oh, I want to go to grad school. I want to study Alzheimer's because I want to save my nana. And that's noble, but those people drop out pretty much instantly, because if what you care about is making an impact on the world, it's hard to sustain the energy when you're, it's 10 p.m. on a Saturday night and you're in the cold room and you're making your 14th attempt to purify the alpha beta subunit of something that maybe in 40 years might somehow be involved in Alzheimer's treatment. That's what research is like. It's hard, and it's slow, and it's frustrating. You have to have, it's a patience degree. You have to have that internal desire to solve the puzzle in order to get through it. And you have to kind of accept at a core level that this is hard, and there is nothing to be done about it but show up and control what you can control, and try again tomorrow. It's really a professional mindset. The reason I put this book here is The War of Art. It's a book about writing, but writing is also hard, and it's hard in the same sort of way that research is hard. And it really helped me get through grad school and that undergrad research lump. I'm like, oh dear, is this really what I'm going to sign up for? Research is a creative endeavor, and you have to learn how to do that, as once the answers are no longer known and you can't just look them up. And developing your research focus. And then this is the final sort of topic. And my advice for trying to figure out what you want to be studying as a professional researcher, I think my first advice is try not to overthink it, especially if what draws you to research is kind of the puzzle solving and the tool building, and you're a kind of a task-based person like a bioinformatician. You can solve problems in a lot of different ways, and you can expect to have a bit of a randomish walk. Like, I started out with a real plan, and I still my resume can kind of give you the head scratchy sometimes. But I'm still, I'm using my skill set to look at these nasty hairballs of data and turn them into information. And that's been consistent the whole time. But you should be thinking about it to some degree. Like, don't just jump in. Do some thinking. Don't analysis paralysis, but something you should know is your aesthetic. Like what sorts of research makes you want to read the paper, and what sorts of research makes you remember that you've got a hair appointment or something. Like personally, I'm into systems biology, and structural biology is just not for me. It puts me to sleep. I think it's valuable and important, and I'm glad that somebody else is doing it. And that is purely an aesthetic choice, and that is okay. You cannot do all the research in the world. It's okay to pick one, and you should definitely be aware of what your skill set is. Again, I'm not really a hardcore math person. I'm a hardcore pattern detection person, and I make that work for me. And so I am right at the Q&A time. So I've got 2 book recommendations, or 2 reading material recommendations. You should know what impressive looks like, at least in the context of, this is for college admissions, and it's applicable to academia as well. Look up Cal Newport and the failed simulation effect. It's like, hard work is not impressive. Doing something that people can't understand how you did, that is what reads is impressive and what makes people want to give you more opportunities. And as for choosing your research problems, you really want to be looking for things that are adjacent possible, like N+1 problems. Like developing a new method from scratch is really, really hard. You have to respect that. Say, okay, here's my tool set, and if I just added this one thing, what other problems could I solve? I heartily recommend this essay on it from Dr. Hamming, You and Your Research. You can look that one up. Okay, and now it is time for me to stop talking and open the floor for questions.
Thank you so much, Dr. Aull. That was such a fascinating talk. I enjoyed the book recommendations and also mentioning the war of art, because I think one of the stereotypes about STEM is that you don't really need as many of those soft skills, such as like writing, communication skills. When you get up to grad school, you're gonna really need a lot of those skills. So I really appreciated that.
Yeah, I mean, look, if you've done all the work and it never gets beyond your lab notebook, then, as the Mythbuster said, the difference between science and screwing around is writing stuff down, but it's also making sure other people read what you wrote down, because otherwise it doesn't matter.
So in our question and answer, we have a question from Samarth. It says, "Hello, Dr. Aull. I really enjoyed learning about your experiences with the DIY biology movement. Could you share more about those experiences?"
Sure. So I was unemployed and bored. This was 2008, and this website called IO9. It's like a science fiction website. They had a competition for amateur genetic engineering, and they were hyping it up. It's like, oh, grow a tail, and you're like, dude, I do this for a living. You cannot grow a tail. So I decided that I was going to set aside, one unit of monthly rent and my tiny closet, and say, okay, this is what a trained genetic engineer can actually do. And I used like my senior year project at MIT, and I made this little pulse generator, which is like a little circuit, analog circuit part. And by the time the contest was over, I had this little like pale blue E. coli. Like, hey, look, I made a thing out of parts that I could access as an amateur. It was like a rejiggered lambda-phage. It makes a pale blue E. coli that was probably a pulse. And I submitted this, and like, it made the news. And trying to explain this to journalists, I realized that I'd been in the MIT bubble too long and had entirely the wrong idea of how much the average person knows about circuit theory. And that the people who were interested in this were not like, the hardcore nerd contingent, but like, average people. So I developed this genetic test, just like a PCR SNP assay, and because people are interested in their genetic material and they understand that part, and so you could just like do a little SNP specific PCR in your house and do that instead. And so we had like workshops and had a little amateur lab space to do that in. And so that was kind of my outreach learning activity that I did during that time. But then I moved to San Francisco, and I could no longer afford a closet.
Samarth has also asked, what advice can you give to high school students who want to pursue similar innovative projects on their own?
If you have any connections whatsoever, leverage them. If you don't, then what I did is I focused on these sort of science contests in order to kind of build up my credibility and show that I actually knew what I was talking about. And like I said, it is really challenging, and they do have like RSI is great. There are some programs like that, and so I would definitely encourage you to go after those if you can.
Awesome. Another attendee is asking, what are your future plans or goals for your research?
So I am enjoying my job. I'm kind of in this supportive last resort slash algorithms development slash weird stuff make it workers, but it is very much it is very much applied, and part of me does kind of miss like, hey, research questions. So one thing that I'm kind of hoping to do is like have is I'm trying to set aside some of my time to like actually read literature and think about science level questions again. It's true that the difference between industry and academia is like on a day-to-day basis, like what you're actually doing, the very zoomed in problems that you're solving are quite similar. But it is fun to kind of zoom out and say, hey, I'm curing cancer over here. And I'm figuring out something that nobody has seen before, rather than I'm trying to figure out exactly how the customer messed this up because I'm kind of impressed. So yeah, that is definitely a goal on the I'm hoping to spend more time thinking big thoughts, and maybe when the time is right, maybe going to like a startup or something that is really closer to the research side of things at some point in my career.
Awesome. Thank you so much. Another question that I had thought of is, as someone who found their passion for STEM and bio early on in life, what advice would you give to someone who's interested in STEM, like in a loose sense, but is unsure of how to settle on a discipline?
Sure. I think you're not going to figure out what speaks to you by like sitting staring at the wall and thinking about it. You're going to find what speaks to you by going out and trying stuff. Like, I like that's how I found my path when I was like, I'm in a lab. Technically my job is to like, care for all of these cells, but what I want to do is go play with the complicated data that the actual biologists are afraid of. And so it's like, what am I being drawn to? What am I like, I'll trade you so I don't have to do that, and just follow your compass in that way. So I think like listening to yourself and also listening to, some there are practical considerations. I found that the more awesome your job sounds to an 8-year-old, the worse it actually is in practice. Specifically because you know, what your nobody in this world gets what they deserve. They get what they can negotiate. And if there is a line of people out the door who have been dreaming about being, say, like a marine biologist since they were 6, and they would do anything to replace you, you don't have a lot of negotiating power. But if you are willing to, troubleshoot some customer microarrays and you know how to do that, they don't want to fire me. So there's a trade-off. You want to be able to match what speaks to you to what is a rare and a valuable skill that you can use as leverage to generate whatever outcomes you want. And that outcome could be anything. It could be, I want to work part-time. I want to do only the coolest problems out there that totally excite me on a scientific level. I want to make ridiculous amounts of money. You probably can't do all of these things at once, but you're not going to do any of them unless you have something to bring to your side of the negotiation.
Xander is asking, what type of things could stand out to colleges and in general when it comes to the personal projects they pursue?
Read about the failed simulation effect. But the short version is that if I could tell you right now what's going to be impressive, it won't be, because I would know how to get there. What you do is you show up and you learn about opportunities that are only available to the people who have shown up and have built up that credibility. And you can, you're making little baby steps until you're like, wow, how did a 17-year-old end up working on SARS? Or, when I was doing a lot of competing, I was the captain of 5 academic teams. That seems kind of impressive, but like, you know, I got one of them because it was like, I was the captain of the science bowl team and we won nationals. And the oceanography teacher is like, hey, there's an ocean science bowl. And if we win, we get a trip to Hawaii. So I was captain of the ocean science bowl team too. And I had an entire team of people who'd taken the oceanography class, which is not even a thing that exists in most high schools. So my resume, got longer. And again, hey, there's an envirothon and you know how to compete. Why don't you come over here and like be on this envirothon team? And my resume got longer. So again, like it's you build up this virtuous snowball where if you have established yourself as the go-to person for something, or if you've established yourself as an insider, opportunities start coming to you. And it's not something that I can just, prescribe to you in advance how that works, but show up, do a good job and keep your eyes open for opportunities that just aren't available to everybody.
Building on that idea of making connections, maintaining them, and then building on a foundation, what advice can you give on finding a professor during undergraduate research to continue research with?
So we talked about aesthetics, we talked about that sort of thing, but also I think people underrate how important mentorship is. There is a very wide spectrum of how much a professor is going to be interested in the experience of undergrads like you and giving you a good experience and actually bothering to write you a rec letter once you're done and that sort of thing. Like the best advice that I can give you is talk to the people who have worked for them. If you can find one of their old grad students who's recently graduated, they will tell you exactly what's up, because academia has a really wide range of personalities and a really high tolerance for, and frankly, sociopathic behavior. And you do not want one of those, because you're a baby. You need a mentor who takes interest in you and wants to help you get better, rather than somebody who is just kind of stuck there because they're obliged to take students and college makes me I don't want to. Really think about mentorship. Don't just go for the one with the shiniest CV and the longest list of awards, because you're probably not even going to meet that person.
Thank you so much. And thank you all to the audience too for all these great questions we've had come in. We're getting a little tight on time, so I think I'll throw in one last question. If anybody has another one, you could throw it into the question and answer or in the chat. But for Dr. Aull, for you, what do you think the future of biological engineering looks like?
And I think now we have gotten over the hump of the hype cycle, and we are off into we're actually using this, and it's not as transformative as maybe you would have hoped for 20 years ago, but it's people are genuinely making useful, like like algae that are better at producing biofuels in various ways, and doing that in sort of composable and modelable ways, and that's a useful thing that exists. GMOs are still an option. They're difficult politically. It makes me sad. Is really, you need a giant company that's as evil as Monsanto to be pulling off like the sorts of trials that you need, and being able to absorb the damage if some Berkeley students destroy your test plot or something. And so it's harder to have that sort of thing in the environment. And there are also things like the GMO mosquitoes, where they don't actually make they make sterile offspring, so the mosquitoes die off, and those are being released, that sort of thing. Like there are use cases for it, and I think it is now in the, hey, this is a stable technology that exists, but it is certainly not quite as popular as, say, curing cancer, which cancer is one of the few natural predators of congressmen, so it's always going to have funding.
Thank you so much for that. It's just interesting to see, to hear about some of the areas that these students in particular might be engaging with in the future and just hearing what that might look like. So thank you again, Dr. Aull, for presenting with us, and thank you, students, for all those awesome questions. We'd also like to thank our sponsors, including the Claude Moore Foundation, for making all of this possible and providing this. If you'd like to continue attending these events for free, please make sure to sign up and register for our future sessions and to give us a shout out online. And also remember to complete our post-webinar survey. We have a QR code right there, and we'll also drop a link in the chat shortly. Thank you, everybody, for joining us. I'll leave the screen up for a little bit so that you can get the QR code if you need, and then we'll put our link in the chat. Thank you, Dr. Aull, for joining us. It was wonderful. I learned so much, and we look forward to hearing from you again at some point.